one way in which our body obtains energy from macromolecules is by converting triglycerides or fats into acetyl coenzyme A. So basically, if our body hydrolyzes triglycerides into fatty acids, those fatty acids can then be broken down into molecules called acetyl coenzyme A. And the acetyl coenzyme A is fed into the Krebs cycle. Inside the Krebs cycle, it is transformed into water, carbon dioxide, and the energy is obtained that is used to help our body function. So the thing that we're going to focus on in this lecture is the conversion of the most common fatty acid in our body, palmitic acid, into acetyl coenzyme A. So once again, triglycerides are esters that can be hydrolyzed into fatty acids and glycerol. Fatty acids are carboxylic acids that can further be broken down into molecules called acetyl coenzyme A. This acetyl coenzyme A is fed into the Krebs cycle to produce water, carbon dioxide, and energy, and this energy is used to help the body function. Now, the most common fatty acid found in the human body is palmitic acid, whose conjugate base is palmitate. And it's the palmitate molecule that is used to transform it into acetyl coenzyme A that is then placed into the Krebs cycle. And we're going to focus on the reaction mechanism of the conversion of palmitate, the conjugate base to palmitic acid, into acetyl coenzyme A. So let's begin with the first few steps of our reaction. So basically, let's examine step 1, step 2, and step 3. So the entire purpose of the first 3 steps of this reaction is to transform the relatively inactive carboxylate ion group of palmitate into a much more active and reactive molecule, the thiol ester group. So basically, this is the palmitate. We have our oxygen that bears our negative charge, the carbon-oxygen double bond. So we have a resonance-stabilized molecule on which the charge is delocalized among these two oxygens. And we also have the hydrocarbon R group, which basically contains the methyl group on the end attached to 14 CH2 groups. So this R is basically this hydrocarbon group here. Now, in the first step, we take an ATP molecule, adenosine triphosphate, and we react it with this palmitate molecule, and we produce this molecule as shown, which is known as palmityl-AMP, where AMP is adenosine monophosphate. We also produce the pyrophosphate, basically 2 separate phosphate molecules. We have the 2 phosphate groups as well as the oxygens around those phosphorus atoms. So we also produce this molecule, which is basically our conjugate base of palmitic acid. We have the R, the carbon-oxygen double bond, this oxygen, and we have this single monophosphate group. We have our sugar here, and we have our adenosine. So the entire purpose of creating this is to create a good leaving group. So this oxygen is not a very good leaving group, but if we add this AMP molecule onto the oxygen, we create a much better leaving group. We create a strong Lewis acid. Now, at the same exact time, a base reacts with our coenzyme A attached to a sulfur and H. So this acts as the acid, gives the base our H, producing this conjugate base that can now act as a good nucleophile with this strong Lewis acid. So this acts as a nucleophile attacking this carbon, displacing the pi bond, placing it onto our oxygen, and we produce this tetrahedral intermediate that contains 4 different groups attached to this carbon here that we're going to call the central carbon. So in step number 3, we have an elimination reaction taking place. So this is our addition reaction. This is our elimination reaction. So the pi bond is reformed between the oxygen and the carbon. At the same time, we kick off the good leaving group, this AMP molecule, adenosine monophosphate. So in step 3, we produce adenosine monophosphate as well as palmityl-SCoA A. So the entire purpose of steps 1, 2, and 3 was to transform the carboxylate group into a thioester group as shown. We replace the oxygen with our sulfur as well as this coenzyme A. Now coenzyme A is a very large structure. That's why instead of describing the structure by actually drawing out the entire coenzyme A, we're simply going to designate with CoA or coenzyme A. So once we form this molecule, we transform an inactive molecule to a much more reactive molecule, and now the different types of enzymes that exist in the cells of our body can basically help transform the palmityl-S-coenzyme A into acetyl-coenzyme A. So let's look at our final reaction mechanism. So here we have the palmityl-S-coenzyme A. So we basically replace the R group with this group shown here, and this group is here. So we have 1 methyl, 12, 2 more, so 14 groups as described in this section here. Now, the first enzyme that basically helps us through process number 4 is an enzyme called acetyl coenzyme A dehydrogenase. Dehydrogenase simply means the enzyme removes an H atom. So let's see exactly how that works. So instead of actually drawing out the entire enzyme molecule, and this enzyme molecule is very, very large, we only draw out the sections that interact with our molecule here. So basically, this section of our enzyme, acetyl coenzyme dehydrogenase, is a carboxylate anion, and this section is this 6-member ring that contains a nitrogen. So basically, this oxygen here grabs the H, deprotonates the H, leaving this bond. This bond forms a pi bond between these 2 carbons. And this oxygen is kicked off along with these 2 electrons here, and these electrons attack this nitrogen, displacing this pi bond. So the enzyme basically removes 2 H atoms, and we form a pi bond between the beta and our alpha carbon. So this is the alpha carbon, so we remove the alpha hydrogen, and the beta carbon, we remove the beta H atom, and we form the following alkene. So we have an alkene group here, we have the carbonyl group here, as well as this sulfur group here that contains the coenzyme A. Now in step number 5, once again we use an enzyme. So to save space, I'm not going to draw out the actual enzyme and these functional groups on the enzyme that react with our molecule. Instead, I'll simply give you the name of the molecule and what it produces. So we have our enzyme enoyl coenzyme hydrase, which now acts to add an oxygen group onto that molecule. So we basically undergo an oxidation reaction. So we add an H atom onto this carbon, and we add a hydroxyl group onto the beta carbon. So the alpha carbon gains an H atom, while the beta carbon gains a hydroxyl atom, a hydroxyl group. And in the 6th step, another enzyme called beta hydroxyl acetyl coenzyme dehydrogenase now takes off an H atom from our oxygen. So it takes off this H atom, and we also take off this H atom, and a pi bond is formed between this oxygen and this carbon. So we form this intermediate, which is basically our beta keto ester. So now another enzyme acts as a nucleophile and it attacks the sulfur group of the enzyme, attacks this carbon of the carbonyl, displacing the pi bond, placing it onto our oxygen, and in step 6, we form this intermediate. So this is our addition reaction. So once we form the intermediate, an elimination takes place. And this entire group is what is eliminated. This serves as the good leaving group. So when our pi bond between the oxygen and carbon is reformed, as it was reformed in this step, this entire section is kicked off, leaving this section by itself. So we form the pi bond between the oxygen and carbon. We have the S and the enzyme, as well as this hydrocarbon backbone attached to our carbon. And now this molecule can further be broken down, but we're not going to focus on this molecule too much. Instead, we're going to focus on this leaving group. So these two electrons form a bond between this carbon and this carbon, kicking off this pi bond, placing 2 electrons onto our oxygen. And in the next step, in step 9, we take some type of acid and protonate this oxygen to form our enol. In the next 2 steps, we have an equilibrium reaction between the enol and its ketone counterpart. So basically, we take an acid, we protonate this carbon to form this carbocation. In the next step, the base removes the H from the oxygen, and the pi bond is reformed between the oxygen and carbon, and we form this ketone, which is the ketone counterpart to this enol. Now, this is more stable than the enol, so equilibrium will lie towards this molecule, and this molecule is known as the acetyl coenzyme A. So the acetyl is this section, and this is our coenzyme A. So this is the molecule that is now placed into the Krebs cycle, in which it helps transform it into carbon dioxide and water, and we also harvest a certain amount of energy that we can then use to help the different functions, different processes in our body function. So once again, palmitic acid is the most common fatty acid in the human body, and it can be converted into acetyl coenzyme A. And this is what the focus of this lecture was. We basically discussed the organic chemistry, the reaction mechanism of the conversion of palmitic acid into acetyl coenzyme A.